Okay, 3, 2, 1, it's jam.
What's up, science fam? Food is great. All living things need food. Every living thing needs to eat. So the question is, how do plants get food, and how do living things in general get energy from food? These are questions we're going to tackle today. My name is Sir Ramon, and this is Science with Sir Ramon. So our topic for this session is photosynthesis and cellular respiration, all in one go. So buckle in, strap in your seatbelt, and try to survive. So what are we up to? We're going to explain how photosynthetic organisms use light energy to combine carbon dioxide to get energy-rich compounds, describe how organisms get energy through what we call respiration, and recognize that all organisms are energy carrying out all functions required for life. So consider this, a tree, big home. You can just look outside, hopefully you have trees outside your home. Trees have a particular organ that gives them the ability to give shape. So what plant organ is that? You have the leaves. The leaves are wide and flat. They have a certain structure in order to aid in their function, which is to catch sunlight. So they're specialized organs for photosynthesis. Now, what organelle makes leaves green? So this organelle is actually the chloroplast. These are organelles that are specialized for photosynthesis. And they have an inner membrane folded into what we call thylakoids. So they have many thylakoids that increase surface area. So these chloroplasts are key for what we call photosynthesis. So what makes the chloroplast green? What is this pigment that makes them green? It's chlorophyll. It's a pigment in the thylakoid membrane, the skin of those flattened sacs that captures the light. So what is the purpose of photosynthesis? Literally, photosynthesis means photo, light, and synthesis, making or generating or putting things together. So you're using light to put something together. So this is the general chemical equation of photosynthesis. It's a metabolic process that makes food in the form of glucose from carbon dioxide and water. So plants use sunlight in order to be a source of energy for putting together carbon dioxide and water in order to make glucose, a complex molecule. So why are most plants green? What is it about chlorophyll and greenness? When you look at white light or solar light, you have many different wavelengths of light that translate to many different colors of light. Chlorophyll in particular absorbs blue and red light. So if we look at the spectrum here, blue and red light, blue on the left side, red on the right side. What's left is the yellow and greenish light. So what isn't absorbed is reflected. So that's why you see plants as green, because the blue and the red are absorbed. The blue light has high energy, which is exactly what the plants want. And the red light travels farther compared to all other wavelengths of light because it has a very long wavelength. So it lasts until evening. That's why the sunset, you have a red shift or red flash at sunset because red light can travel far, which means that red light lasts the longest throughout the day. So it balances between highest energy and longest light hours throughout the day. So how does light energy get captured by chlorophyll? This is thanks to the light-dependent reaction. So this is phase one of your photosynthesis. So light-dependent reactions basically use light energy to make chemical energy, and it all happens in the thylakoid membrane, the skin of those flattened, folded sacs of the inner membrane of the chloroplast. So the light-dependent reactions, again, in your chloroplast, always looks like a short on the side here, the thylakoid, the flattened sacs, don't like pancake. If you look at the skin of those, we have these things. These are different enzymes and proteins that help us do the light-dependent reaction. And the basic summary is that it harvests energy from light and electrons from water. So we're going to go through this step by step. So we'll focus on that part for now, this little part here. So it says PS2 over there, that's photosystem 2. This is a special group of proteins that catches and processes light. So again, a photosystem catches and processes light. So step one, what happens is that light excites a chlorophyll electron. The light comes in here and excites an electron in a chlorophyll molecule. So that excited electron, that energetic electron, it's sent away to the next step and next reaction in the light-dependent reaction. But since that chlorophyll molecule sent away an excited electron, it's missing one electron. We need to replace that missing electron. And how the photosystem does that is that water is broken down to replace the missing electron. So the energy from light itself is used to break down water in this chemical equation. So water is done as oxygen, some hydrogen ions, and some electrons. So this is called hydrolysis. So water is being broken down using light energy. Okay, so again, we break down water to get the electrons and replace the excited electron that was sent away because it was too excited by light. So that is photosystem 2. You might be wondering what photosystem 1 is. So oxygen gas is basically just a waste product of hydrolysis. Let's go back to that. So again, it's just a waste product. It doesn't do anything in this light-dependent reaction. It's just there. What we needed from the water are the electrons to replace the missing electrons. Right? So that's the first part. Now we're going to move on to this middle part here. So that middle part is what we call the electron transport chain for photosynthesis. So there's proteins between what we call the photosystem 2 and the photosystem 1. It's called photosystem 2 even though it comes first because it was discovered later than photosystem 1. Right? Not get confused. It's just, by order of discovery, not by order of how it functions. So, going back, step 4. Remember the excited electron that was sent away from photosystem 2? It goes into the electron transport chain, and the main point of this is that the excitedness, the energy from that hot electron will be harvested in the electron transport. So the electron gives that solar energy to pump hydrogen ions into the chloroplast, or into the inner membrane. So it pumps the hydrogen ions, and that pumping gives energy to make ATP, or adenosine triphosphate. This is the energy of the cell. So again, we now have energy in the form of ATP because of the energy from sunlight that excited the electron in photosynthesis. Okay, so again, summary is, in the first part, we have an excited electron. In the second part, we use that excited electron to make ATP. Right? So now we are in the last stretch for the light-dependent reaction. This is photosystem 1. So again, it was discovered first, but it functions later in the light-dependent reaction. So this one starts off with, again, exciting of chlorophyll electrons. So light can also excite an electron of the chlorophyll molecule here, and that excited electron is sent away as well. But the special thing here is that instead of hydrolysis, the electron that replaces the missing electron comes from the electron transport chain. So you see this arrow here, that's the electron transport chain, the one that's low energy. That's going to be the one that's used to replace the missing electron. Now the excited electron gets carried off by NADP plus or gets recycled into ETC. So there's two options that can happen. So this excited electron can either go forward in what we call the non-cyclic flow, or it can go back to the electron transport chain. The one we discussed there, it can go back to create a cyclic flow. So what's the point of this? There's two things that can happen to that excited electron. The cyclic flow helps with the chloroplast generate more ATP, more energy, while the non-cyclic flow, the one that makes it go forward to NADP plus, generates more NADPH. Now, NADPH is what we call an electron transporter. It's a taxi, it's a tricycle that just takes electrons from one place to another. So in other words, cyclic flow makes more energy. Non-cyclic flow uses the electrons and yeets it somewhere else. It's to generate electrons. So again, energy and electrons. So that's the main product of your light-dependent reaction. So then the main product of your light-dependent reactions, we have ATP, or energy, so that's from the electron transport chain over here, and NADPH, which carries electrons. So we have that over here. So again, these are the things that are made in the light-dependent reaction. These are ingredients for the second phase of photosynthesis, right? You with me? So again, first phase, light-dependent reactions. We use solar energy to make ATP, chemical energy, and NADPH, electrons, in a convenient package, right? So in the light-independent reaction, this is the second phase of photosynthesis. It doesn't need light, but it can still function in light. So again, it's just independent. It's whether light's there or not, it can happen. It's also known as a Calvin cycle. So this happens in the stroma, the gelatinous matrix, with the soup inside the chloroplast. So the Calvin cycle has three main phases. We have carbon fixation, we have reduction, and we have regeneration. We're not going to tackle the specific molecules, but we do have to tackle what these three stages are. All right? Let's go through them one by one. First step, carbon fixation. From the term itself, we're fixing carbon from carbon dioxide into a compound. Let's see what's happening here. So when we stop here, we can see the carbon dioxide from the atmosphere being fixed into a different compound. So coming from here, we have 1, 2, 3, 4, 5 blue circles. So those blue circles are carbon atoms. Again, those 5 carbon atoms, you add 1 carbon atom from the carbon dioxide, and it becomes 6 carbon atoms in that one compound. So again, that's just basically carbon fixation. You're adding the carbon from carbon dioxide into a compound. And this is a key part of photosynthesis, and it's made possible by rubisco. It's a very important enzyme. Without it, blackout, we know it wouldn't exist. So after carbon fixation, we have what we call reduction. So reduction is basically the term for gaining of electrons. You're shoving electrons into that compound. So electrons from NADPH, which we got from the light-dependent reaction, and energy from ATP, which we also got from earlier, will be used to make G3P, or glyceraldehyde 3-phosphate. But let's just remember G3P, because G3P is used to make glucose, right? So again, reduction in this background here, we have ATP and NADPH. We're just shoving energy and electrons into that molecule to make G3P. Or we have an energy-rich molecule that's used to make glucose. So 2 G3Ps can be used to make 1 glucose. But for every turn of the cycle, only 1 G3P will be satisfied from making glucose in each cycle. Why? All the other G3Ps that are made in each turn of the cycle will be used for the last step, the last stage in the Calvin cycle, regeneration. Or if it's a cycle, you need to reset that cycle. So some of the G3P, 5 of them to be exact, will need to reset the cycle. So we need energy from ATP in order to recalibrate those 5 G3Ps into 3 of the starting compounds earlier. So again, you're just using energy to rearrange everything and reset the cycle. So again, carbon fixation, add some carbon dioxide to your carbon compound. Reduction, you add electron energy. And regeneration, we're just resetting the cycle. Right? So the product here is G3P, which is used to make glucose. So sometimes rubisco doesn't exactly do what it's supposed to do. So rubisco sometimes actually accidentally starts the opposite of photosynthesis, called that photorespiration. So when rubisco binds with oxygen instead of carbon dioxide, so instead of putting a carbon into the compound, it binds with oxygen and does the opposite of what it's supposed to do, in waste. So in order to help with that, some species of plants have different photosynthetic pathways. These are different ways of doing photosynthesis. So first, going back to the Calvin cycle, the normal Calvin cycle, how many carbons are in the first stable compound? If you count the blue circles, you have 1, 2, 3. For each one, it's 3. So this is where we get the name for the normal pathway, the most common pathway, T3 pathway. It means that 3 carbons in the pathway. So it happens in just how we discussed it. Basically, we just have one cell. There's no new adaptations. In the same cell, we have the light-dependent and light-independent reactions happening in one cell. That's the T3 pathway. Rubisco, though, binds to oxygen even more. So it wastes more of our time at higher temperatures, like in desert or in tropical areas. So it's best to have some other way to do things. So that's why there's also the C4 pathway. So instead of a 3 carbon compound, we have a 4 carbon. So minimizes photo respiration by separating the processes into different cells. So here's how it works. In C4 pathway, carbon fixation, the one with the rubisco thing, is done in a different cell using a different enzyme. So if we look here in the methcyl cell, we have a different enzyme that's attaching a carbon from carbon dioxide to a different molecule for a 4 carbon molecule. You see those blue circles there? 1, 2, 3, 4. So 4 carbons. So that's where it gets the C4 pathway name. And then, so again, that's carboxylase. It doesn't bind with oxygen. That's why instead of starting with this, so we start with a different enzyme that can do the job without binding to oxygen. But this is different from what Rubisco does. So you still need Rubisco later on. So the Calvin cycle continues in a different cell. We call it the bundle sheath cell. When the bundle sheath cell is dropped, the Calvin cycle continues as normal, and Rubisco is not exposed to oxygen at all. It doesn't have to work with oxygen because instead it works with a different carbon compound. It doesn't have to bind to carbon dioxide anymore. Just work with that new C4 compound, right? So the C4 pathway costs extra energy in order to transport it between the cells, in order to work with carboxylate, the other enzymes, but it's worth it in the warmer places, tropical places, because in this way, Rubisco has no choice but to bind with carbon dioxide, not with oxygen, right? So it's still worth it, even if it costs more energy, but only in the warm. So going to the extremes, we have the desert plants. The challenges they face include low amount of water, high temperature, so they need a special kind of photosynthetic pathway. And normally you need more water during the day. So what they do is what we call the CAM pathway. It stands for the crassulacean acid metabolism pathway. Basically, what it does is that instead of separating the processes by different cells, it separates the processes by time. So it's separated by night and day. So during the day, we have pores called stomata that are closed so that it doesn't release water. But they open at night so that they can exchange gases with the atmosphere without losing as much water. Smart planet. So at night, it does carbon fixation while the stomata are open. So again, they're open so they can exchange with the atmosphere for carbon dioxide, so they can do carbon fixation, and it functions similar to the C4 pathway. It makes a 4-carbon compound. All right, so that's at night. During the day, it does the rest of the Calvin cycle with the stomata closed. So they're closed, you can't get more carbon dioxide, but your carbon dioxide has been fixated or stored like a storage bin in your C4 compound during the night. So during the day, it's like just chipping away what it's stored overnight. So it doesn't need stomata to be open, so there's no water coming out of those pores. Very efficient. So like C4, it also keeps oxygen away from the business. So, double function. It keeps oxygen away and it keeps water inside the desert plant. So that's the CAM pathway. So, photosynthesis is complicated, so we don't have to call plants simple. But we're not stopping here, of course. We also still have the other process. So why do we breathe? What is oxygen even for? These are questions that can be answered when we discuss cellular respiration. Now, if you look at the chemical equation, we start with glucose, we combine it with oxygen, and we get carbon dioxide, water, and energy. This is basically the opposite of photosynthesis in the general chemical equation. But the steps are not just the reverse, right? So the steps are, they do a completely different way, right? So even though it looks like it's just, flipping the chemical equation, the process of the chemical equation is not the inverse of photosynthesis. So cellular respiration is its own one. So you'll see that in a bit. So respiration is basically the breakdown of biomolecules like glucose to make ATP or chemical energy. So what organelle is responsible for this? It's the mitochondria, the powerhouse of the cell. So these are some of the parts that describe some of these parts. We have the matrix, the fluid inside. We have the cristae. These are the folds of the inner membrane, just like the thylakoids from the chloroplast. There's folds here, but in this case they're called cristae. There's no folds. They're not flattened stacks. And just like the chloroplast, there's an inner and outer membrane. So we have some similarities with the chloroplast and the mitochondria. So why fold up the inner membrane into cristae? Why have folds? Same as earlier, more surface area. There's more space in order to do its function. And the structure relates to the function. But wait, we have to get the stuff. We have to get the goods into the mitochondria first. And the problem with that is that glucose is kind of big. So first, we find glucose in the inner material of the cell, the squishy part of the cell. We call that the cytoplasm. So that's where it all starts. Your respiration process starts in the cytoplasm, fluid of the cell. So in the cytoplasm, we have glucose. He's a very big boy, looks like the apple chip, and you can't just shove that the apple chip through the membrane to the mitochondria. No, we have to break it up. So in order to break it up, we have the first step is cellular respiration, glycolysis. So we'll do this step by step, just like earlier. So focus. One, in glycolysis, literally breaking the glucose. It's the breakdown of glucose in the cytoplasm of the cell. And this is the simple version we have in my case. Glucose gets broken down into tyrosine. And you have ATP and you have NADH being made from the background. But, we're hardcore, so we're not going to go to the simple version. We're doing basically on this side. Alright? So I guess I'm not really making memorize the molecules, don't worry, but you do need to count that. So first, there are two phases, the energy investment phase and the energy payoff phase. What does this mean? You gotta give before you get stuff. Right? So in the energy investment phase, you give energy. You use ATP in order to make glucose unstable. So if you look here, we have ATP being added to glucose, and this makes it unstable so that it breaks apart on its own. So, instead of you need to exert energy in order to make stuff happen. So you exert energy to make it break apart, and this broken apart thing can be used later in the energy payoff phase. So that broken apart thing becomes G3P. The same molecule earlier used to make glucose in photosynthesis. We're back at G3P. Very important molecule. So in the energy payoff phase, that G3P will get processed in order to get the energy back, plus other stuff. So first, electrons are taken from G3P by an electron transporter called NAD+. So this is different from photosynthesis earlier. Photosynthesis had NADP+, and it became NADP. So just remember photosynthesis, they start with a P, the NADP has a P in it. Whereas in respiration, there's no P. Correct? So it's just NAD plus and NADH. Alright. So in the step number 2, phosphate is taken from G3P to make more ATP. So again, we just take electrons, we take phosphate in order to get electrons and energy from G3P. And the remaining compound at the end is tyrosine, which is very important in the later stages. So again, in the energy payoff phase, what happens is that we get 2 ATP for each G3P, right? So earlier when we broke apart the glucose, we got 2 G3P. So for each one, we get 2 ATP. 2 times 2, we get 4. We have 4 ATP, right? So we gave 2 and got 4 back, so we earned 2 ATP. So how are electrons taken or given? So let's talk about NAD plus and NADH. So electrons get transferred to what we call redox reaction. So oxidation means losing electrons, reduction means gaining electrons. So we have this in the bottom layer here, lose electrons, oxidation, gain electrons, reduction. Another thing is oil rate. We use oil rate, oxidation is more, reduction is gain. So again, remember those. So what happens with NAD plus and NAD plus is that it's like a taxi. The electrons get in, they pay the fare using H plus ion, and then they get transported somewhere else. Basically what's happening with the redox reaction. You gain electrons from somewhere, you lose them somewhere else. Now, what about transferring phosphate? So earlier we had electrons, now we have phosphate. So we have two types of phosphorylation or transferring phosphate. We have what we call substrate level phosphorylation. That's just transferring phosphate from one molecule to another. So earlier in glycolysis, when we got phosphate from G3P and put it on ADP to make ATP, that's called substrate level phosphorylation. It came from another molecule. Oxidative phosphorylation is when you oxidize them, when you use oxidation. So that's the transfer of electrons in order to have phosphate transferred to a different molecule. We'll see that later. So back to glycolysis, the next photo is that we have 2 pyruvates, 2 NADH, and 2 ATP from just one glucose. So, starting at step 1, there's already a lot of stuff. We have 2 pyruvates, which is what's for the next step, 2 electron transporters, and 2 ATP, straight up energy one. Right? So that's just step number 1. So now we have pyruvate, a nice 3-carbon compound. It's very convenient to shoved into the mitochondria. Now we need to do step number two, the transition stage. We need to prepare that pyruvate for what's going to happen next, right? In the transition stage, we're modifying pyruvate so that we can get a lot of oxidation out of it, meaning we're going to take all of its electrons, or most of its electrons. So in order to do that, we need to step it, and we do this through the transition stage. So this is what happens. It happens in the mitochondrial matrix, the fluid is the mitochondria. Basically, you have pyruvate here, and it gets processed by what we call CoA FH. So just remember that this CoA attaches to the pyruvate, but shoves off the carbon dioxide. So we use one of the blue circles here, and we attach COA in order to make what we call acetyl CoA. Now in the process, there's also some electrons that get taken off while the CO2 is being removed. So NAD plus can be turned into NADH. So the electron transport takes electrons from the pyruvate. So in the end, what we have is acetyl CoA, waste carbon dioxide, so this is where the carbon dioxide comes from, and NADH. So in total, we have two acetyl CoA, two NADH for electron transport, and two carbon dioxide as waste, just from that one glucose earlier. Okay? Now we're on to step three, the Krebs cycle. This is also complicated, but we're not going to discuss the names of the molecules, just process once again. So the Krebs cycle oxidizes or takes electrons from acetyl CoA. That makes a lot of electron transporters, which will be used in the last step of respiration. So again, it still happens in the matrix, the fluid of the mitochondria. So again, it's complicated. There's a lot of stuff, there's a lot of molecules, but again, we'll just focus on the process of the product. And so bear with me here. We have here a nice little diagram. So in step number one, okay, if we're looking at the carbon atoms, the blue circles, in step number one, we start out with a molecule that has four carbon atoms. We count this one, 2, 3, 4. Okay, that's four carbon atoms. We attach two carbon atoms. So 4 plus 2 is 6. We get a 6-carbon compound, and so on. So in the next two steps, steps 2 and 3, all that's happening, that's the movement is that we're removing carbon dioxide. Just like what we did in the transition stage, we remove carbon dioxide so that we can steal some electrons from the compound. So removal of carbon, we steal some electrons and make NADH, and we have waste carbon dioxide. In the later stages, steps 4, 5, and 6, we steal some phosphate and some more electrons from it. So in step 4, we steal a phosphate. Step 5 and 6, we steal more electrons. So that at the very end, our product in total, we have 6 NADH for electron transport, 2 FADH2, it's another electron transporter, another taxi, and 2 ATP straight up energy molecules, all from stripping the acetyl CoA, stripping that cute little molecule here that we prepped earlier from pyruvate. We strip all of that, all of those electrons and that energy from the acetyl CoA. Now, all of those electron transporters from the glycolysis and transition stage and Krebs cycle, all of them will go to the last stage, what we call the electron transport chain. So we have all the electron transporters from all the previous steps, and we might then go to the electron transport chain, which is very similar to the one in photosynthesis, right? So it's used to make ATP. So the type of phosphorylation here is what we discussed earlier, oxidative phosphorylation, because it comes from oxidizing or taking electrons from the electron transport. All right? So, high energy excited electrons from the electron transporters take this energy to make ATP. So again, we have NADH and FADH2. These are your two types of electron transporters, and they give electrons, high energy electrons, to the transport chain, which uses that energy to pump out hydrogen ions into the intramembrane space. Now, these hydrogen ions are so packed in the intramembrane space, they flow back in to the matrix of the mitochondria, and when they flow in, the only way for them to flow in through a hole in a special protein called ATP synthase. And when they pass through ATP synthase, it powers a reaction that allows us to make ATP from ADP. So again, the flow of hydrogen ions powers the process of making more ATP. In other words, there's just a factory where we use excited electrons with electron transporters to make ATP. So wait, okay, so there's two types of electron transporters. Now they have different work, different equivalents of ATP. NADH is more efficient, so it can make 3 ATP. FADH2 can only make 2 ATP. Okay, so just remember that one of them is more efficient than the other. Now, at the very end, we have electrons that are just freely going around in the matrix of the mitochondria. They're not allowed to stay there, or else the mitochondria will become very electrified or implode on itself. So we need a final electron acceptor, and the acceptor here in this case is oxygen. So that's why we need to breathe. We don't breathe, our mitochondria explode. So the oxygen accepts the electron and becomes water in the very end. So again, the electrons are accepted by oxygen gas and becomes water. So electrons need to be taken away at the end, that's why we have oxygen. So this is also called aerobic respiration. It requires oxygen. Oxygen is the final acceptor and produces water as a byproduct. Now, if there's aerobic, which requires oxygen, there's also anaerobic respiration. These are types of respiration that doesn't require oxygen. So under molecule similar to the final acceptor, just like sulfide, sulfate. So these are useful in oxygen for environments like in the bottom of the sea. So at hydrothermal vents, we have specialized organisms that don't use oxygen, they use other things that come from the vent. Okay. Another way of getting energy from energy-rich molecules is fermentation. This is a shortened version of respiration. It's less efficient, but also they don't need to depend on oxygen or any other final acceptor molecule. So one type is lactic acid fermentation. So it basically works like glycolysis, but pyruvate, instead of getting into a transition stage, it's just reduced, meaning we just add electrons to it in order to make lactic acid. So again, we just shove the waste electrons onto it and it becomes lactic acid, then it's done. So one example of this is yogurt. Yogurt is made through lactic acid fermentation. Actually, original yogurt from milk, extra yogurt, or even the pork. We also have lactobacillus. So lactobacillus is the bacteria that's there. Next is ethanol fermentation. This is very similar. It's glycolysis, but instead of pyruvate, it turns into ethanol and carbon dioxide. So again, we have a different waste product. In this case, we have yeast that can make bread. So that's why you have to make sure your yeast is alive so that you can make bread that's risen, fluffy. So yeast is also used to make alcoholic beverages through the same process, ethanol fermentation. So photosynthesis respiration is quite similar, I'd say, but what goes around comes around. Your photosynthesis is used to make energy-rich molecules, or energy-rich molecules must be processed into actual chemical energy. And so you don't just expect to get them, you have to do it as well, just like in glycolysis. So, maybe you can apply that to that. Who knows? Anyway, this has been photosynthesis and respiration. I'll see you guys.
Party over.